Okay, you can see today's talk is about biochemistry. So this is the chemistry of living things. And it's a little different, or it's a subsection of chemistry as a whole, because the molecules are generally related to, just a minute, I have to go back. I have to start again. There's a button I have to press. So, okay. Oh gosh. I have to mute you. I can hear someone's radio. So, all right. Now, so let's start. The word matter is an old word, is what chemistry is about. It's about everything and what it's made up of. So we say that all matter is composed of one or more of. Now, so we have what are called the elements, 92 elements. And these are the different atoms, different types of atoms. And elements, so you have oxygen is a pure oxygen, it's a pure element. If you have a piece of copper, 100% copper, it's a pure element. And there are 92 of them found in nature, but they're put into particular order when they're listed. I'll show you in a moment. As well as that, up to now, an extra 26 elements have been man-made, and they're included as well. So there are 118 elements, and they'll probably find more eventually, or produce more. And back in 1869, a fellow called Dmitry Mendeleev, very, very famous Russian, he came up with this idea of, even before he knew how these elements are structured internally, he worked out somehow, and I really don't understand how, or don't know how, is a way of setting them out in a table. And this table is called a periodic table because they are in columns, and each column is which I'll show you in a sec, is called a period. So the information as it was, and from 1869, it wasn't anything like it is today, but the structure, the basic structure was there, and we just kept adding it to this table. So and the information and the new elements were just added continuously. So it's quite a complicated thing today. So this is a, if you go on the net, go on Google and put in periodic table, you'll end up with a whole stack of basically the same, but people have come up with different ideas of actually doing them. I like this one because it's clear. Now, if you have a look, the periods go across 1 through to 8. 1, 2, you jump these. Sorry. So you got the periods down there, the 8 of them. This group here, the blue group, are all metals, and they're called transition metals. I'm not going to go into any of this in detail. Then on top of that, there are 2 other groups here. The lanthanoids and actinoids, and they're all part of it. If you have a look at each of these boxes, here's one for oxygen, which is over here. Oxygen is number 8. So atomic number is 8. Now I'll explain how these numbers come up, how they fit in. So you have an atomic number, you have an atomic weight or a mass weight, and you have a symbol for it. So they all have a symbol and a name. And if you look at other tables on the net, if you like, it's got more information. But if you've not done chemistry before, it won't make any difference. So let's have a look. This number one in group, so period 1, in this particular group here, each one of those is a group as well, but green is very specific. It's a gas. These are gases, and it's not an metallic. So it's a number 1. So it's got 1 proton and 1 electron, and that's it. Very, very simple. Now, number 2 is underneath it. Sorry, number 2 is here, lithium, so helium on the other side. And I'll come back to why you've got these in 1, 2, 3, 4, 5, 6, 7, 8. It all makes a lot of sense. And I'm going to do this, 1, to teach you a bit of chemistry, but 2, so you will understand how the biological molecules fit together in a particular shape, and you will understand that, I'm sure. Okay, I put this in, not to bamboozle you, but just to show you how some people have come up with incredible ideas. So each one of these has got a picture, which is what it's used for. So here you are, silver is a silver pot. Let's see, a chlorine here is, you can, what is it? I can't even tell what the thing, oh, it looks like a soldier walking through it, but they used chlorine during the First World War to kill soldiers. It's a gas. These are all gases here. Anyway, you've got lead down here, so you've got lead as in a bullet. All right, so little things like that. But here's mercury, Hg, is a thermometer in a patient's mouth. So it's a cute little thing. And I just put it there to show that there's a lot of nice stuff around. And there's the thing here about Dmitry Mendeleev. So 1834, he died in 1907. So that's a pretty good age for that time. So here's the definition. It's the smallest particle, an atom is the smallest particle of any element that still has the properties of that element. And that's called the atom. Okay, so if you have an atom of hydrogen or copper or lead or iron or mercury, whatever it might be, helium, one single atom will have the properties of that particular element. So you can have a whole stack of that element. If you've got a piece of gold, as you can see this calculator, if that is pure gold, every little atom in there would have exactly the same properties as the whole thing, by definition. So they're made up, each atom is made up of smaller particles, and this is the guts of it. So what we have, protons, neutrons, and electrons. Now the protons and electrons come together and make up the nucleus. That's where the mass, the weight of the atom is. And around this nucleus, you get spinning electrons, and they spin pretty fast. And they spin around the nucleus. And if you have 1 or 2, that's easy. Hydrogen, helium, they just spin around, 1 or 2 of them. But don't forget things like uranium and 92. So you can't fit 92 in a little orbit. So there are layers of orbits, so just to make sure that they all fit in nicely. So the protons and neutrons themselves are made up of smaller particles. And there are 36 of these particles that make up the neutrons and the protons, including what are called antiparticles. Now, in that dot point, what I've given you here is stuff I didn't do at school. Didn't know anything about, well, even at university, I've got my honors in a science course, and we didn't do anything like this. And antiparticles, I'm still having trouble understanding them, but they're there. So we have other particles like photons. Now, photons are the particles that make up light. Bosons, quarks, neutrinos, these are the names of these particles. And I do not know what they do. I did not have the time to follow it up. So this is a diagram how people used to think atoms were structured. So you had that everybody knew there was a heavy nucleus in the middle, and the protons and the neutrons here, and around them flew the electrons, but not quite as it's shown here. It's more like this. They're in layers. So this is a sample of a carbon atom. Now, carbon is number 6. So it's got 6 electrons. So in this case, it's 2 in the first shell. And these layers are called shells. And 2 and 4 in the next shell. Okay. Now, if you have a look, well, on this diagram, we've got the electrons have a little negative sign. You can see it here. The protons have a positive sign, and the neutrons do not. They're neutral. Now, most atoms that we come in touch with, that we understand we do, the number of electrons and the number of protons is, if they're equal, the atom is said to have to be neutral. 6 pluses and 6 minuses is 0. So there's no charge on these. Then if they do have a charge, they are called ions. They've become ionic. And that's very important too. Okay. So protons have a positive charge, electrons are negative, neutrons are neutral, no charge. And the number of protons are equal. So atoms have a neutral charge. Okay, that's just so important to understand that bit. All right, here's another one. Helium. All right, so that it's got an atomic number of 2, and because there are 2 protons, it's got a mass number of 4. So you add up the number of protons and the number of neutrons. 2 plus 2 is 4. Now, the mass number is a number that's given in how heavy the atom is. Okay, so this is, I only put this in just to reinforce that little bit of theory. Here's a carbon. This is really a useful one because we are made mostly of carbon. So carbon is got a mass number of 12. Why? It's got 6 protons and 6 neutrons, and you add them up, it's 12, so that's it there. However, if you have a look at this, in what's called the atomic weight, it's not really exactly 12. And as I said, there are other bits and pieces, other particles. And they've worked, this is a more modern, relative to when you and I were at school. And that's the atomic weight. This is what's called the mass number. It's a number, but this is the actual weight of the atom. Okay. So if we have the old style, we have the 6 electrons around there, 6 protons, that means neutral. This group of protons and neutrons gives you the weight, the mass of the individual atom, and these other things just move around fast. What is important here is that they are in an orbit, but the orbit is not like in a circle. It's if you could go in and have a look at an atom, they'd be like a cloud. They're moving very, very fast, close to the speed of sound, and speed of light, and they are just spinning through at a particular level above the nucleus, which is this mob here. They're the nucleus, and then you've got 5 different layers as the atoms get bigger. Now, as they get bigger, they get heavier, not because of the electrons, but with what's in the nucleus. And as they get bigger, the outer what are called the shells, and they're actually labeled, and don't ask me why, but they're called KLMNO. And I say, go figure. There may be no reason, but I mean, I've known about this for a long, long time. They've known it for a long time. But I've never read anywhere or seen anywhere or heard anywhere why they came up with KLM and so on. All right. So these shells are important. So the electrons are arranged in these specific shells. There are 5 possible shells, as I mentioned, and the shells have the same number of electrons as the number of protons. So we know it's going to be a neutral atom, and that's so far so good. Now, the first shell, these are the 5 shells, and the first shell, the inside one, is K, and it's got a maximum, can hold a maximum of 2 electrons. The L can hold 8, the M 18, N 32, and O 50. And if you add that up, 50 and 32 is 82, 92, 100. That adds up to 100, and that's 110. Now, there are more atoms than that, so there are other ways of fitting them in, but that's the basics. So in a bigger atom, you have, don't worry about counting the numbers, but you have these shells, and the shells are really like a cloud within a cloud within a cloud. The outer ones, the outer 3, have layers of cloud. So the KL, say the M, will have a couple of layers of cloud, M, KLM, KLMN, and O have many layers, because if you have 92 electrons, say for uranium, which is the heaviest of the natural ones, if trying to fit 92 electrons around a nucleus, how do you do it? Well, you fit them in these shells, but they have different energy levels, and so they're actually layered. I think that's the best way to explain it. I hope you can get with these diagrams an idea of that. Okay, now we talk about the elements, the atoms. Now let's look at molecules. Now, if you join by definition, 2 or more atoms joined together gives us a molecule by definition. So water, for example, I'm not sure what's next. So water, for example, has H2O, as everybody knows. And so it's got 3 atoms. So that's a molecule of water. hydrogen is H2, oxygen is O2. So they are actually molecules, not atoms. They are made of atoms, but by definition, because there's two or more of them, they're called molecules. So I'll be using all these words later. This is really the introduction for those who haven't done chemistry for a while or ever. So water has got three atoms, two hydrogen and an oxygen. And this is how they're spread out. Now, I'll try and explain this a little bit more detail than I had before. Now, the way this is done, oxygen has let's remember the period 8 on the right-hand column, all those are neutral, very, very, very stable elements because they have full outer shells. Now, in the remember Kl has to have 8. So oxygen being number 8, anyway, it has 6 in its outer shell. So K has got 2, and because it's number 8, but so take 2 from 8, leave 6. But that second shell, L, can take 8. So 2 of those extra atoms, electrons, come from these hydrogens. So there's a hydrogen, which has only got 1 electron, actually shares that electron with the oxygen. So these are 4 of the, remember, it's got 6 in the outer shell, in the L shell. So the oxygen has got 6, 2, 4, and then 2 more. So one of those 2 shares with this hydrogen, the other one shares with this hydrogen. So then it's got an 8 in the L shell, in this case it's the outer shell, and it's a very happy, very stable molecule because it's got a full outer shell. It's got 8. 2, 4, 6, 8. Now, you're wondering why are they drawn differently? These 4 here are the spare electrons. They're not bonding with any other atom. These lines here refer to a bond, and each bond has 2 electrons because in this case they're shared. So if you add there's 2 there, 2 there, that's 4, and the other 4 are 8. So and hydrogen really needs 2 electrons to be stable in the K shell. K shell can take 2 max. So one of the electrons from the oxygen, one of the electrons from the hydrogen make this 2. So you've got 2 going around this hydrogen, 2 going around this hydrogen, and 8, because 2 of them are shared, going around. Does that make sense? No. Okay, you nodded. Thank you. Here's another one, a little bit more complex, oxygen. Now, oxygen is also very stable, and oxygen is number 6. So it's got 2, 3, 4, 5, 6. Hang on, 1, 2, 3, 4, 5, 6 in the outer shell. So sorry, 8. So what happens here is the 2 oxygens are sharing from each 2. So there are 8 altogether around them. Okay, so they're sharing, and because they're sharing 4, 2 from each. So those 2 are from this oxygen, there 2 here. So there's 4 there, another 4 there, it's 8. So it's the same number as this around this oxygen, but in a different configuration. But the interesting thing here is, because they're the same element, in this case, this is what's called a double bond. It's a very strong bond. It's much easier to break a single bond than a double bond. Okay, so it's just the way the electrons are being shared.
Ruben?
Yes?
I have a question.
Sure.
So that makes sense, the sharing makes sense, but when the electrons are spinning around the nucleus,
Yes.
and it's being shared, how
Yes.
does that happen? It goes around the nucleus?
Yeah, they fit around, yeah, but don't forget, they're moving terribly quickly.
Yeah.
Very fast. So they actually move within the shells of both. They just move right around. So but the numbers are there, okay, the number of electrons, and that keeps them stable.
Okay.
So if they lose for let's see, now oxygen, for example, if there's only one atom of oxygen, but it finds two more electrons, and I won't go into how it finds it, but if it does, it'll have its 8 electrons there, but the 2 extra ones are 2 more than the protons. So that has a charge of 2 minus, 2 negatives. The electrons that we say are negatively charged, and the protons are positively charged. So if you have 8 positives and 8 negatives, it's neutral. But if you have 8, say,
electrons.
So yeah, I'm losing it. So if you have, say, six protons and 8 electrons around it. Oh, actually, this is Basil. Sorry. Hi, Basil. Oh, yes, I've been going for about half an hour already. Oh, I sent it to you. I sent it to everybody. That was 2 weeks ago. I sent one on Tuesday, Sunday. Go back. Yeah, I think the Sunday morning. I look forward to seeing you. Yeah, bye. He didn't see my email. I'll go on anyway. So there are different ways of atoms that can attach to each other, and where I have these, so that was a bond. If I was to draw this one, it would show 2 lines, one above the other. That's called a double bond. I mentioned that before, I think. Okay, so these bonds, I would hold them together, but they're not solid. It's just the electrons that make them up move so fast. If you could take a snapshot of them, you wouldn't see anything because they're so small, but they're very, and that's, these electrons moving fast hold us, people, all matter, the computer, everything's held together by this electron spin. Now, I mentioned these two, the first one anyway. So the 2 types of bonds that we look at, all right, we'll go a little bit deeper, just a little bit deeper. I don't want to scare anybody off. We have these ionic bonds I mentioned there. They have a charge, a positive charge or a negative charge. And then we have the covalent bonds, which have no charge. So they have the right numbers of protons and electrons. The ionic bonds do it differently. So the ionic bonds, when there's this sharing situation, so electrons of 2 or more atoms move to fill each other's shells. An example. Okay, so the atoms are stable. They don't break apart. They don't do anything. You're just happy. when the outer shells are filled to capacity. So if L shell can take 8, if it's got 8, oh, what more can an atom ask for? They're happy. Everything's really good. And substances with ionic bonds dissolve very, very easily in water. So, for example, if you have something like salt, sodium and chloride, they share electrons. And because of that, in water, they just fall apart. Because then what they do, they break away, and then water molecules are attracted to each of those. And don't forget, the water has got a positive end and a negative end. So it loves ionic bonds. So here's an example: sodium chloride. Sodium has is number 11. So it's got 2 in the middle, K shell, 8 in the L shell, and 1 in the M shell. So it's in the first period. It's got 1 in the outer shell. Chloride is sort of the same, but the M shell is missing 1. Okay, so it's got 2, 4, 6, 7. Now the M shell is made up of more than 1 layer. So, but let's just say the first one can only take 8, but it's missing 1. And so the 1 from the sodium M shell pops in here, and by it moving to here, from the sodium to the chloride, the sodium is now very happy because it's got a full outer shell, not the L shell. On the other hand, by having picked up this extra electron, as you see over here, this has a full outer shell. So they're both happy. And so long as they're together, it's a very, very stable element. Compound. It's a compound. It's a molecule. Hey, Basil. Gosh. All right. Okay, I'm recording it, so I will send it to you. You can follow it up later. Is that okay? Bye. So the idea of ionic means that they form easily and they break up easily. So like salt. And now, hopefully, you understand the idea of full outer shells. It gives the atoms stability, but because they're sharing these electrons, in this case 1 electron, they have a certain fragility about them as well. So if there's no water around, you can get salts, you can get cake salt, whatever, it's quite hard. As soon as you add water, they split and move away from each other. The other sort of bond is the covalent bond, and they actually are sharing electrons in a certain way. And sharing them to actually fill the shells. And these shells are difficult. Now, I should mention, just go back to this one. When they say the sharing, they are together, but that particular electron is going between the two. They're sharing it in time, but they don't just sit there. These electrons are moving the whole time. And so this one thinks it's got a full outer shell, and this one thinks it's got a full outer shell. But in reality, that electron is just moving between the two. Does that make sense? maybe. Okay. Whereas covalent bonds, they actually do a share, and the electrons stay there, and they're very, very stable bonds. So they're very stable molecules. They're very hard to break. Those. So hydrogen chloride, or hydrochloric acid, if you like. So hydrogen's got one atom, just one in the K shell. With chlorine has 7 in the outer shell. So if this one and this one come together, they share the electron. So the hydrogen's got 2 moving around its K shell, and the chloride's got now 8 moving around its L shell. Happy as Larry, everything's good. Hydrogen chloride is very, very stable. So these ones here are examples of very stable covalent bonded molecules. On the right here, there's water. So if you have a look, hydrogen, like here, hydrogen's got 1. This X would be the 1 electron for the hydrogen, here and here. Oxygen has got 2, 4, 6. Hang on. No, sorry, 2, 4, 6 in the outer shell. It needs 2 more. So the 2 from the hydrogen are there, and these electrons sort of float very fast around the 3 things. And because water is extremely stable, and it takes a lot of energy to separate them, that's where boiling comes in. What boiling does, they don't break apart, they actually break apart from each other. So steam is just individual molecules moving around. As soon as it cools down a bit, they just join together again. They're attracted together. Ammonia is the same thing. Nitrogen has got, if you have a look, 2, 4, 6, 7. That's not right. That's 5 there. So I'm counting these K shells. I shouldn't be doing that. The outer one, L, 2 there and 3 more, 5. And so 1, 2, 3 makes 8. That makes sense? Okay. Methane, carbon has got 4 in the outer, and therefore it can take 4 other electrons, in this case hydrogen. That's called methane, CH4. Carbon dioxide is a very stable one too. Now oxygen so as you see, carbon and oxygen here, they have enough electrons there to give themselves stable, and these are all covalent. All right, so in this case, if you count the pink ones around the, this is oxygen. So they each have they share 4 electrons between them, and to make them stable. So they each have 6 on the outer shell, L shell. All right. So water, oxygen, carbon dioxide are what are called simple inorganic molecules. Now that once we start getting into the living molecules of living things, we call them organic. So carbohydrates, fats, proteins, nucleic acids like DNA and RNA, they are organic. They're not like water, they're not like carbon dioxide, which are tiny molecules. These are massive molecules with hundreds of thousands of atoms. So energy, this is really important. Without energy, we can't do much at all. So all cellular, okay, and therefore bodily activities require energy. If we don't have energy, we will be put underground. Now, glucose is a main source of energy. Everything that we eat can be converted in the body to glucose. And we've got in all cells except the red blood cells, these little gadgets here called mitochondria. One mitochondria, ION, and multiple mitochondria. And they make adenosine triphosphate. So it's adenosine in the molecule, and it's got 3 phosphates attached. Now, the bonds of the phosphates are what are broken, especially the end one, and that's what gives us the energy. I'm moving my hands about, I'm talking, I'm thinking, I'm breathing, and the same with you, except for the talking bit. And whatever we do, it's ATP that does it. That's how our muscles move within each other. They slide within each other. Our hearts don't stop. Hopefully, this is all got to do with this ATP, adenosine triphosphate. It's not a small molecule. It's not a big one either. But it supplies us with energy. Okay, so all living things, with a couple of exceptions, use that. Now, where do we get our energy from? Carbohydrates. Now, if you look at the word carbohydrates, it's made up of carb, as in carbon, oxygen, O, and hydrogen. That's what they're made up of. So when the body runs low on carbs, on carbohydrates, then we can actually convert fats around the body, and which will then be producing ATP. And when the body runs out of fat, then the body's proteins are broken down to ATP. Because without ATP, we're dead. So when there's that, when people just don't have food for days on end, you start converting fats, but weeks on end, you'll be converting proteins, and you just fade away to a skeleton. So here's ATP, and this is an amazing little invention by somebody. Now, the adenosine, the ATP, so this is adenosine, and there are 3 phosphates. There's a molecule called adenine, which is almost identical to that, which is part of the DNA making process. But I won't go there now. So it has 3 phosphates, PO4, 1, 2, 3, 4. But it's missing, each one of these 4 are missing a hydrogen atom. So that's why there's a negative charge there, and there. But the bond here attached this phosphate to this oxygen is what's called a high energy bond. So as whatever we do, whether we're thinking or we're talking or we're running or we're cooking, whatever, this molecule fuels it all. Everything that we do is this molecule. And this bond here, it's usually drawn as a squiggly line, like a tilde on the keyboard. But that's fine. You've got a big yellow star there. That's when it breaks, energy is released. The broken bond, it releases the energy. And that's used to do whatever has to be done. Now, these mitochondria look like this, basically. There are 2 layers of membranes, and they've got ribosomes, they've got DNA, because the current hypothesis is that mitochondria used to be some form of very, very early organism. We're talking about billions of years ago. And when other cells formed, that had energy issues, and the mitochondria were taken up by these other very primordial cells. And then you had this wonderful relationship where one was given protection The mitochondria was given protection by the other bigger cell, but the bigger cell was given energy produced by this mitochondrion. One ION is singular. And that's why mitochondria have their own DNA. So each of us have got two types of DNA in our bodies. We have a cellular DNA, which is in our normal cells in the nucleus, and we have mitochondrial DNA. And of course, the mitochondria and the mitochondrial DNA are passed through to each of us, each new baby, if you like, from the mother. The father, the sperm, only carries the cellular nucleus, which is what does everything else. But the mother's egg, for example, will have a number of these mitochondria and produce cell for the growing cell, and they just keep reproducing them. And the other interesting, I should say, so if you're into genealogy, they talk about where you came from. So whatever part of the world you came from, the DNA is slightly different. So through your mother's line, they can trace where you're probably originated from, or at least your mother's line originated from. Okay, so that's why they talk about mitochondrial DNA, mDNA. And the other thing I should mention, depending on how active you are, the number of mitochondria in muscle cells change. So if you're sitting around most of the day, you have enough mitochondria there just to keep you going. But if you all of a sudden you start walking every day and you start walking really fast every day, the number of mitochondria are increased. So it's got its own DNA. When energy is required, more mitochondria will be produced. Okay. So carbohydrates, all our energy source is really from glucose, and that's what it looks like basically. I just explain it briefly. It's all made up of carbon, hydrogen, and oxygen. That's what's called carbohydrates, I mentioned before. Now, when you draw a diagram like this is shown in 3 dimensions. So the thicker line is towards us, the thinner line is at the back. So you've got the hydrogen, oxygen attached to carbon, and so you get basically that. Glucose is hexagonal, fructose is pentagonal. They're similar, but they're different. And so here's sucrose. Sucrose is a combination. Now, sucrose is the name for sugar, normal table sugar, what you would put in your tea and coffee or bake with. So it's got a glucose and a fructose together. And there's just an oxygen there, which they share. Don't forget, oxygen needs two electrons from 2 different molecules to share to become stable. And if you look at this, so back here, there's C6H12O6, all right? And here, see, you've got 6 carbons, this one's got 10 hydrogens and 5 oxygens. So they're the formulas for those 2. Now, carbohydrates are divided into 4 types. monosaccharides. Mono means one, that's one sugar. Saccharide is a sugar. Disaccharides, two sugars. Oligosaccharides are anything up to a molecule up to 100 sugars. And over 100 sugars in the molecule are polysaccharides. Starch are very complex the carbohydrates, and they're made of sugars. And then we've got fibers, and like as in your diet fibers, they are also very complex carbohydrates. They're not as simple as those. And if I lose anybody with a definition or you don't understand, just stop me and I'll come back. I'll get back to something. Now, photosynthesis is a process that where plants use light to produce their food, but they use very simple molecules. They take carbon dioxide, you saw before CO2, carbon with two oxygens, that's all. Water, you saw before, H2O, and light. And together, they produce glucose, C6H12O6. So they take carbon dioxide and water, and through, it's almost like a magic spell, you get the sugar in the chloroplasts. Now the chloroplasts are similar to the mitochondria I showed you, it all happens in there, but they need light. So glucose is then used to produce everything else. We can produce every molecule in our body from glucose molecules, everything. Now, and we store glucose, glucose can be stored as starch, and that's a very complicated one. We store glucose in our liver, but not as starch, a similar molecule, but it's very big. This is how photosynthesis is. This is the formula for it. So you need 6 carbon dioxides from the air. This is in plants, green plants, usually, mostly. 6 waters, and they produce the C6H12O6, that's glucose, and there's your oxygen, which is rubbish. It doesn't need it. So it goes into the air, and it wasn't until there was enough oxygen in the air in the very early days of the Earth could other organisms start to exist, because they couldn't produce their own oxygen. They had to breathe it in or absorb it in the air. So plants, very, very early, algae and so on, in water, produced over millions of years enough oxygen to cover the Earth, and then the next level started, and then we ended up with us. But that's going back a while. So for this to actually occur, it needs an enzyme of some sort. And there's a molecule which is not quite an enzyme, but it acts as an enzyme, called chlorophyll. And by the way, chlorophyll comes in different colors. So most plants that you see are green, but you can get it red as well, and get yellow as well. But green is the normal one. And so glucose is a precursor to the other sugars. So everything in the body starts up with glucose, which is an amazing molecule if you think about it. Now, when plants are eaten by animals, most of it's digested, broken down to glucose. So the plant will eat the sheep will eat the grass, chew it, and it'll just break down the glucose. Then the glucose is used to produce the energy in the mitochondria, as I showed you before. But this case now, I've given you a little bit extra, ATP, and that's this molecule with adenosine with 3 phosphates on it. And between the second and the third phosphate, remember, it's called the high energy bond. So whatever you do, that's the one that gets broken off. If you're a long distance runner and you use up all these high energy bonds, then the second one, the next one in, will start to break up. And so it's all pretty good. Now, this process is called cellular respiration. The other one's called photosynthesis, and it's exactly the reverse. So if you look back, 6 carbon dioxides and 6 waters giving us glucose and oxygen is the reverse of this cellular respiration. So what we do to get our ATP, our energy, is the reverse of what the plants do. Now, fats are composed, that was carbohydrates. Fats are composed of very long chains of carbon, oxygen, and hydrogen. They're the same atoms, but a different structure. So you've heard of saturated fats and unsaturated and monosaturated. And you have saturated fats where every bond, every possible bond is used to put a hydrogen on. All right? There are no double bonds in there. Whereas here, there's a double bond. So it's not saturated anymore because there are, up here, if there was a single bond, you'd have 2 more hydrogens. And so this is called an unsaturated. You've all heard of these words, these names. And because it's only 1 double bond there, it's called monounsaturated. Okay, so if there are only single bonds, it's saturated. If there's at least 1 double bond, it's unsaturated, and with 1 bond, it's with monounsaturated, diunsaturated, polyunsaturated. If more than 2, okay, even if more than 1. The polyunsaturated margarine is what people like to buy because it's much, much healthier than saturated. Now, if you eat animal fat, the fat that you eat from animals is saturated, and that causes problems around the heart. Then we have triglycerides, and about 95% here of all dietary fat are the triglycerides. And then animal fats and vegetable fats, they also contain triglycerides. And once they're in the circulation, the body can actually use them for energy. And that's fine. But the problem is, if we have too much of it, we store it in body fat, and which is okay for small amounts. But if you have large amounts, you've got issues with that. Then, if you have high blood triglycerides, so is a sign of conditions that the heart is at risk. There's a risk of stroke. There's a problem with obesity, and you can get a thing called metabolic syndrome. Now, the metabolic syndrome is a cluster, it's a group of conditions which includes too much fat around the waist. And when that's usually the sign, then blood pressure is high, triglycerides are high, blood sugar is high, and you get a very high, abnormally high cholesterol levels. And that is where you'll be seeing a cardiologist. or at least a position in that area. So this is what triglycerides look like. This group here in this rectangle, that's called glycerol, and attached to the glycerol are three bonds here, and you can have any different sort of fats, and depending on what the three fats are there, they could be all the same. You can have three different ones, or two and one, whatever. You get a different triglyceride, and some are worse than others. So this one's showing an 18, a 14, and a 16. This is just in the diagram as an example. And these are the things that cause. You can see here, there are two hydrogens missing there and here. So this one is saturated. Those two are unsaturated. So depending on the combination of those fats, you get different types of fats. So what have we got? We've got saturated fats, trans fats, monounsaturated, and polyunsaturated. I'm sure you've all heard of these words, yes? No? Maybe? So saturated fats and trans fats are regarded as the bad fats, because they're the ones that will produce like a layer inside the arteries in the heart, where you need stents put in. Unsaturated fats, is said the good fats, they're the ones everybody wants. So just to revise, saturated fats, there are no double bonds. That means all hydrogen, maximum number of hydrogens can be put on. Now, there are two sorts of unsaturated fats. There's what's called the trans, where these hydrogens here are opposite each other, or the cis, where they're on the same side. This one here, the trans, is the worst of the lot. They're not allowed to use trans fats in Australia in making food. In America, they do it. In America, they have much more higher level of heart issues, and they're putting a lot of it down to having trans fats in their food. And just dependent on the positioning of this bond with the opposite ways that the hydrogens are facing, it's causing that major problem. So bad cholesterol is LDL, low density lipoproteins. Lipo, by the way, is fat and protein, so it's a combination of a fatty protein that carry cholesterol particles through the body. Okay, so it's a combination of fat and protein. It's bad because it builds up these deposits inside the walls of arteries, and then as they get harder, thicker and thicker, they get harder and harder. They lose their elasticity, for one thing, and also it reduces the annulus, the diameter, so the amount of blood that can go through that effect, and that can lead, and little bits of the fat can break off, end up somewhere in the brain or who knows where, and that causes problems like strokes, and so that the heart, the narrow, and the risk of heart attack. The good cholesterol, the high density lipoproteins, HDLs, they break up that, so they pick up the excess cholesterol and take them to the liver, where the liver breaks the whole thing down, gets it out of the system. Now, having a high HDL is linked to lower risk of heart disease, heart attack, and stroke. Okay. Depending on how much animal fat you eat, you end up with a bad LDLs, low density lipoproteins. And if the fat that you eat is mostly vegetable, plant-based, it's much, much better.
Ruben, can I just ask you? I'm a little
Sure.
confused. Why is it called the low density, which is bad cholesterol? And why is it high density when it's good cholesterol? What is the density standing for? Of what? Of the fats?
Of the molecule.
Of the molecule.
Yeah, yeah.
So the more there is of the fat, there should be the bad cholesterol.
The more, if you eat a lot of animal fat, you eat a lot of let me think what's the word, saturated fats.
I see.
And because they're saturated, they're called low density. Whereas here, the good cholesterol, it's a protein, but with a fat attached, so it can transport it. And so the good cholesterol is mostly with the hydrogens there, and without the double bonds.
Yeah, just wondering why the density low and high seems to be.
I'm not 100% sure, but I think it's got to do with what other atoms are attached to it.
Oh, I see. Possibly. Thank you.
Yeah. But you're okay with the idea of the good and the bad, the high density and the low density?
Oh, yeah. Well, I understand. I just thought it should be the opposite. The low density should be the.
No. So if the fat that you use to cook with or that you eat is basically is plant based, it'll be high density.
Yeah. Oh, yes. I see.
Okay. So here, this is basically it. That the high density, you see, I found this is a new one. I found a new diagram. So high density liver proteins and low density. The high density with the good one has got minimal cholesterol, if you have a look at that, whereas the low density got a lot of cholesterol. So the proteins, these blue ones, are the proteins that seem to reduce the amount of cholesterol in them as well. And the triglycerides, there are fewer of them in the high density than the low density. So there's your cholesterol there, and there are fewer of them there than here. This is, as far as I understand, this is basically what they look like in reality, just a diagrammatic form. Okay, now, so the triglycerides and cholesterols and other essential fatty acids. By the way, I haven't mentioned this yet. We have to have certain fats that we can't make, because don't forget, things like the brain is mostly fat with all sorts of neuron connections, but very fatty. We have all our nerves right through are coated with a layer of fat. It's called myelin sheath, and that stops the nerves, the signal going through the nerves, being lost to the water in the body. So these fats here, they store energy, they insulate us, they protect our vital organs. They act as messengers. A lot of the hormones are made from the fats. A lot of the hormones are also made of protein, of course, but they do act as they are from fats. So they start chemical reactions that these hormones, like they control growth, immune functions, reproduction, and other parts of metabolism. So we are made up of trillions of cells, and they all have a cell membrane, and the cell membrane has got cholesterol within it. So we must have cholesterol in our diet. but not too much. It's the excess that causes the problem. So, that's it. There's no harm in eating it. You need it. Let's look at proteins. Now, fats and sugars are made up of carbon, oxygen, and hydrogen. Add nitrogen, and you get protein. And a couple of the amino acids in proteins also have sulfur. What happened here? So we have 20 different amino acids naturally occurring in our bodies. And if we eat meat or any protein, whether plant or otherwise, or animal, this is what we get in these 20. The shape and function of a protein. Now, this is the important bit. There are 20 different amino acids, and every protein has a different sequence of the proteins, the way they're laid together, end to end. And the sequence of these amino acids determines the shape and the function of the protein. So this table, I don't expect you to know it. These are 20 amino acids that we have. These are the normal letters. We don't use the second. This is just a clear table that I found. Ignore the third column. Now, proteins have, like everything else, have bonds, and these, when the bonds are holding two amino acids together, they are called peptide bonds. Peptide bonds. All right? Two amino acids together then, which are held together by a peptide bond, they are dipeptide. If you've got up to 100 amino acids, it's called a polypeptide, or just peptides, or polypeptides. Any more than 100 amino acids, they're called proteins. Okay, definition, that's all it is. Now, this is what an amino acid looks like. It's called an amino acid because it's got this NH2 group here. Now, normally, if you write this amino group molecule on piece of paper, you'd write an NH2. But because it's the N, the nitrogen that's linked to the carbon, it's written this way. And so a carbon, you remember, it's got 4 electrons in its outer shell. So it can have 4, or it will need 4 other electrons to stabilize it. So 1 electron comes from this nitrogen. You get a single bond there. Hydrogen has a shared electron there. This carbon will share an electron there. And the R group is, which will make all of these together. It's a separate small molecule, which when added to the basic structure of the protein, to the amino acid. So this bit, the hydrogen and the carboxyl group here, is standard in all amino acids, and the R group is one of the other bits, the 20. Now, the acid bit is here. That's the amino bit. The acid, the carboxyl, the oxygen and hydrogen, is a single bond there. And when that's broken off, when the hydrogen is moved, the hydrogen is the acid part. So, how do we make a protein? Here you have your NH2, your CH here, the COOH. Now, one of the hydrogens from the next door here, oh, by the way, it's R1 and R2, that just means 2 different groups. It doesn't have to be different, but it can be. So here, this hydrogen is broken off from this nitrogen. Okay, so this one on the left here, the amino group. This oxygen and hydrogen are broken off here. And together you get an oxygen and two hydrogens. You get water. And you get a bond here between the nitrogen and the carbon, and you get what's called a dipeptide. Di means two. You get two peptides, or you get two amino acids together with a peptide bond. Now, because you're producing water, it's called a condensation reaction. Yep. So proteins are very large. There are thousands of amino acids joined together, and so they produce quite a bit of water as well. So we have the capacity in our DNA, because that's where the sequence of the amino acids for each protein are stored. We can produce up to about 100,000 different proteins, not at the same time, but we've got the capacity from as soon as a zygote, an egg and a sperm come together and make a zygote, the very first cell, they start producing proteins. And as an embryo grows and the fetus grows and then the baby grows and we grow, and what we produce changes for depending on our needs at the time of our age. Okay, so that's all part of the 100,000. So the things that we produce in utero, once we're out and they're not needed anymore, because the blood for example, in a baby in uterus is somewhat different to adult blood. So that stops being produced, that difference. So some of these amino acids have a positive charge, some have a negative charge, and some are neutral, they have no charge. So the sequence of these amino acids will give us the shape of the protein that's produced. And it's the shape which will determine its function. So here's a fairly simple looking example. This is insulin. When insulin is produced, it's produced in a much bigger molecule than this. What you see here and what you see here are the same things, just a different diagram. And each one of these are the table of 20 amino acids, and that's where they are here. Now, what you see here are sulfur bonds, cysteine. This cysteine and that cysteine there, and the same here and the same here. Don't worry about the outer one. The two inner cysteine and sulfur bonds for glucose to enter our cells. They a part of this molecule is sort of at the edge, sticking out a little bit of the cell, and it works like a click-clack model. I don't know if you remember, we had these models when we were kids, and they're a click-clack, 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 just change. But what happens here, I'll show you with my hands. It opens up a glucose molecule will pop in and will change it to that. And the molecule then goes into the cell. As soon as the molecule of glucose is out of the place, out of this molecule, out of the glucose, out of the insulin molecule, it pops back. So the insulin molecule does that. It's click-clack, backwards and forwards. And that's what these sulfur bonds are for. So this breaks and the molecule and the glucose comes in. As soon as the glucose molecule is in here, something triggers it, this closes, and this one opens, and that pops it into the cell. Ingenious. Does that make sense, guys?
Mm.
Yeah. Now here's a protein molecule. There are different sorts of proteins. There's some that are called the alpha helix, and they actually, you'll see it in the blue here. If you follow them, and there are different sorts of, well, we talked about bonds before between, so some of these have little straight lines, and some of the bonding is what's called hydrogen bonding, the very weak bonds. So the hydrogen and the oxygen here, they're attracting each other, but they're not actually, it's not a strong bond. So that shape is called an alpha helix. These are called pleated sheets. And this is quite what the proteins look like. And depending on where they are in the body, well, they do have different functions. So computerized modeling now looks a bit like this diagram here. So, so it would, this was the alpha helix, that's what it looks like. And this is what the sheets look like. In the hemoglobin, heme means iron, and globin means protein. Hemoglobin is protein carrying iron. So that's a complex, it's a complicated molecule with an iron atom in the middle, which is then attracts and holds oxygen. So that a hemoglobin, that's all, if you like it, it's called a heme molecule. The hemoglobin overall is like this. So that's not hemoglobin, that's the heme bit, which is here. And that's, so it's got, how should I say it? So the hemoglobin molecule is made up of 4 proteins, 2 alphas and 2 betas. And in fact, you can see it here. There's alpha 1, alpha 2, and up the top beta 1 and beta 2. Embryonic hemoglobin has got the alphas like adults do, but they don't have the betas. They got another version called the gamma. And then as the baby is being born, or in the birth process, the body of the baby stops making gamma and starts producing beta. So not long after birth, the whole thing is exchanged to have adult, the baby will have adult blood. So within each of these proteins is a heme group. This is what attracts the iron. And this is what they look like. So I don't know which one's the alpha, which is a beta, but that's there. And then within each one is the iron aspect. Now, with proteins, we have quite a few different sorts of proteins that we make. We have hormones which control, they communicate, and there's internal communication. Some animals, of course, produce hormones to communicate with others. For example, a lot of insects, the female will produce a hormone which she pumps out into the air when she's ready to mate. It's like moths and butterflies. And they follow the scent of the female of their species to them, and first come, first served. We produce enzymes. So anabolic, the ones that build up molecules, the catabolic that break them down, and others that produce energy. We have structural proteins, so keratin. We have keratin, right, our skin is layered with keratin, our hair, our fingernails, horses' hooves, they're all keratin. They're all protective, they're all fairly tough proteins. Collagen is what gives us our shape. So the collagen in our face is there and is produced. We have contractile proteins called muscles. We have defensive proteins, antibodies. We transport things around the place. hemoglobin, mentioned that. By the way, heme means blood, globin, protein. I mentioned it before. We have receptor proteins, part of the communication system within the body. Signal from the brain goes somewhere else, it's picked up by a receptor. Very specific. We don't have storage albumin, but eggs are, the albumin in eggs are proteins. So they store energy, and they store, there's enough there to build a new organism inside the egg. And the last lot I want to look at is nucleic acids. Now, these are ones that produce DNA and RNA, and we call them the bases. And there are four, well, there are actually 5 of them, but 4 in DNA and 4 in RNA, slight difference. And then what we have here is, you've heard of DNA, of course, but we have what's called a nucleotide. So we have a nitrogen base, which is, I'll get to in a second. So that's it there. There's a sugar, a 5-sugar, and a phosphate molecule. So we've seen these before. This is a bit different. Now, so the building blocks of our DNA and the RNA, the very slight differences between them, but they're totally different in function. And and so these are called nucleotides. You have to have these 3 molecules there, and then I'll show you different ways that they're linked. So DNA, if you look at this, take one of these nucleotides, and there's the base. The C is for cytosine. There are 4 bases in DNA and 4 in RNA. Cytosine and guanine. Now they are, I don't know if you can see these little red dotted lines here. They are, if you like, hydrogen bonds, and they're quite weak bonds. So they can break fairly easily once the whole thing's unraveled. So we have 4 of these, C and G and A and T, adenine and thymine, and the various combinations of it. So these hydrogen bonds are what keep them together. So if you then, if you like, this is the sugar. I'd like you to notice that on one side, if you like, the peak bit is pointing in one direction, on the other side it's pointing down. So even though they seem to be parallel like a ladder, they're running in opposite directions. So they're called anti-parallel. And so this is sort of the equivalent of, well, the first 4 of that. And this is what the double helix looks like. I won't go into the history of the discovery of the structure of the double helix. I don't have the time today, but it's very, very interesting. So of the human genome, we have about 20,000 genes which code for our proteins. But we can actually produce up to about 100,000 genes, 100,000 proteins. Now, each gene is composed of the nucleotides, which are these. So one of these is a nucleotide. And there are only 4 choices here. So it's the sequences that are important. And the sequence or the order of these is unique for every protein. If it wasn't unique, they'd all be the same, which we know is not the case. Okay, so this is a part of the gene. And there's a section there called the promoter, and that's where it gives instructions to the process of producing the start of the production of the protein. And each section has got two things called exons and introns. All right, now we have the first process, which is called transcription. Now, if you remember back when we were in primary school, the teacher would have written up a sentence or a paragraph on the board in chalk, and we copied it absolutely perfectly down. That is transcription. You're copying exactly. It's an old word, but it's used in genetics. So transcription is when the DNA is copied exactly into RNA. And there are, I go into differences of DNA and RNA when we talk about genetics itself. So I won't go into now. And then, so these are exactly the same, but in a different format. And then there's a splicing program, process, where the red bits, if you like, the introns are chopped out and the exons are merged together. They're glued together, if you like. And this is the bit of the gene that actually codes for the protein. Then the translation happens outside the cell, and an amino acid is produced according to the sequence along here, along the messenger RNA. And then once the amino acid chain is done, it folds itself into the actual protein. Now, I said there are about 20,000 genes, but you can produce more than that, many tens of thousands more proteins in our bodies. So if you think about this as a very simple example, the blue ones here, the exons, or here if you like, are the ones with the actual code, where the code is stored. Now, if you take the four of them together, you get that. If you leave one of them out on purpose, or two of them out, or put them in a different order, you get a totally different protein. So depending on what protein is required, what happens in this process here could be different for each gene. I hope I haven't lost you, but that's a fairly new idea. When I was studying at university, we were told we've got about 100,000 proteins, we've got about 100,000 genes. In 2003, the Human Genome Project, the very first one, well, there were two, they came out a few weeks apart. One was government, one was privately run. And I said, we've got 35,000 genes. And then as time went on, the way they counted, the way they worked out what is a gene, it's somewhere between 20 and 22,000. I'll just say 20. I don't know exactly what the number is. They keep changing it because the definitions change slightly. But we still have the same number of proteins. We haven't changed, just the way they count the genes. Now, you've seen this before. I just go through it again. It's quite straightforward. This gray line here with the spaces, that's the wall of a nucleus. So the nucleus is in there. The DNA, what should I say? Okay, so the DNA here is open. The system wants to produce a particular protein. That particular gene that will do that protein is found, and this molecule notice it's called the RNA polymerase, and ASE on the end means it's a protein. It's a protein. It's an enzyme. It's an enzyme. A polymer, it means many things. So many amino acids in this case. Or anyway, so this polymerase will separate the DNA strand here and produce what's called a messenger RNA, this blue thing. These nucleotides are RNA. They're a little bit different from the DNA ones. They also have three. Anyway, I won't go there. No. So this blue thing here, it pops out through one of these gaps. And the green thing at the back is another form of RNA. And it's called a ribosome. And it's on this. So this thing moves through like in a factory. And there's another sort of RNA here called tRNA, transfer RNA, and it brings in amino acids, and they wait around till a specific code here is on 3 of these, and they pop them on, and this pink thing is the protein. It's called the polypeptide chain. Peptide is the name of the bond between 2 amino acids. So many of those. And this is called transcription, where an exact copy of the section of the gene is made, and translation is is where you've got the translation into a protein. Okay, and this is protein synthesis. It might look complicated, but it really, once you look at it, it's not too bad. So here's a simplified one. DNA, remove the so you got this is insulin. So that is formed. It's a pro insulin, and this is pro insulin. So it's been chopped around a bit, and it and with insulin, because remember it's two like a ladder. So it comes out like this, and then the orange bits in between, the whole thing folds over like that, and the orange bits are cut off, and you end up with that. And there your 2 sulfide bonds. Okay, I've got to put that in, and I think you might have an understanding, because so that originally the insulin molecule is originally a straight protein, and the system bends it, they attract each other, you get the section of it cut away, goes away, it's reused somewhere else for something, or chopped up with amino acids, and then you get that. And ladies and gentlemen, we're done. And I hope you got something out of that. An idea of, that's called biochemistry. The chemistry of life.
Ruben, can I just ask you, with the new vaccines, some have mRNA and some don't, messenger. I'm just wondering what this is about, as far as putting, which is better to get with a messenger RNA or not?
It is. What happens is the messenger RNA, well, the messenger RNA takes the code, and the system then produces the protein of that messenger RNA code.
All right.
So what they have done with COVID, the coronavirus, a section of a spike, they worked out a sequence, made messenger RNA out of that. And the messenger RNA in our system then goes into individual cells. Each cell that has a bit of this mRNA will then pick it up, make the protein of that they wanted us to have, of a section of the of a spike. It goes into our system, and then our immune system does not recognize it. So then that goes into overdrive to work out what needs, what needs to be done to get rid of it. So if we are infected in a later time with real coronavirus, the spikes are there. Now, unless the new coronavirus comes along and the spike protein is different, if it is different, then the mRNA vaccine is useless. But protein viruses don't change their spikes much at all. And so it's a very good way of doing it.
Mm-hmm.
Does that make
because
sense?
I had the vaccine of Pfizer, I became very ill. So
but that's not necessarily the mRNA bit. It's whatever it was in, whatever the liquid had in it. You reacted to it.
Yeah, I see.
Yeah, I've had, my wife and I, we both had we had Pfizer, we had all the various, we haven't had any problems. So your system reacted.
So I should have had another injection, a different one. Thank you.
Yeah. Thank you, Preeti.
Thank you.
Yeah. I just read your message. Yeah.
It was a great class.
Thank you.
Thank you so much.
I hope all that wasn't overwhelming. I've simplified it as much as I could without losing anything.
Excellent. Thanks.
I don't know if any of you've got a chemistry background, but no.
Oddly enough, I have the chemistry background, but I haven't done it for years. So it came back. Thank you.
Well, things have changed. All right, guys. I will see you in a fortnight.
Thank you so much.
Thank you. Bye, everyone. Thank you.
Be well, stay well.
Bye-bye.
The virus is still busy out there. Bye-bye.
Yeah, bye-bye. See you.